Hello everyone, I'm really happy to talk to you today, taking a little break from the field with my students. And I'd like to use this opportunity to tell you a little bit about my lab, the Macrophysiology lab. I'm Maria Thaker and I'm an Associate Professor at the Center for Ecological Sciences at the Indian Institute of Science. If you ask me what my lab is all about, I think the best way for me to describe is to use this phrase. We're interested in phenotypes. And we like to take an integrative approach to understanding animal form and function. And what this means is that we want to know how bodies are built from the inside out using things like physiology, and from the outside in understanding the ecological context. And so for the next 5 minutes or so, instead of telling you 8 years worth of research that we've done so far, what I'll do is I will organize this into two research themes that have dominated the kinds of questions that we've asked over the years. The first is our research in phenotypic shifts in a changing world. In order for us to understand how phenotypes are shifted, we've been trying to capture these changes as the environment has been changing. And so to do this, we have a couple of simple steps we follow. First, we find a beautiful lizard. And so here are three examples. The superb fan-throated lizard, Sarda superba, the Indian rock agama, Samophilus dorsalis, and the Mysore day gecko, Nemaspis mysorensis. These three species have been model systems for us where we started with them and we built from it. And they all share a really interesting characteristic. They all are beautiful, which means they actually have extravagant color that is extra and above what is needed for survival. We then find an ecological context to compare. And we do this because ecological differences are a great way for us to capture differences in selection pressure. So for example, we've been comparing urban versus rural habitats. We've been comparing wind turbine versus non-wind turbine areas, or closed versus open forests. We then measure phenotypic trait expressions and the potential trade-offs between these trait expressions. So, for example, we've been measuring morphology in terms of body form and color, or physiology, circulating hormone levels, or thermoregulation. We've also been measuring behavior, like social behavior or anti-predator behavior or movement behavior in their landscape. And finally, we connect these phenotypic trait complexes to the selection pressures that we think are operating across the different spatial contexts that we compare, or across time when we compare species across evolutionary time. So over the last 8 years or so, we found ourselves studying these two selection pressures more than any others, sexual selection and predation risk, and often the interplay between the two. Right? And we've asked questions like this. How do hormones, colors, and behaviors interact to influence the social outcome? What are the costs and benefits of these elaborate signaling traits? For the predation risk kinds of questions, we've been asking, how does predation risk itself change trait expressions? And how does nutrition or nutritional ecology influence the phenotypic trait expression under fearful conditions? We've even started studying multi-species interactions. So for example, Sitana laticeps and Sarda darwini both live in the same area, which means they both experience the same ecological conditions. And by studying how they interact with each other, we can better understand competition between species and whether that's a way of shaping phenotypes within species. And our newest research direction, which we're very excited about, is thermal adaptation. This research program now allows us to study a different axis of physiology that we hadn't so far, and it allows us to capture environmental heterogeneity and species strategies in order to deal with the challenge of environmental heterogeneity. So, for example, we've begun now comparing urban and rural habitats in how heterogeneous they are and how species are thermoregulating within this landscape. This takes us to our next research theme, which is animal movement ecology. In order for animals to, to maximize benefit, minimize cost, which means get as much food and as much reproduction as possible while suffering the least cost as possible, they've got to move efficiently through their environment. And so far, we've been studying animal movement ecology by tagging and tracking mammals across their landscape. So the image right here is a golden jackal with one of the colors that we've put on it. And we ask questions like this. Right? How do mesocarnivores like golden jackals or jungle cats or Indian foxes coexist with each other? And how do they do that in human-modified landscapes such as agriculture? What physiological and behavioral strategies can be used to solve the ecological challenges? Right? Is there a way for them to utilize human-modified landscapes and still stay away from us while gaining the best benefits? Right? And finally, are there generalities in movement strategies across sites? So if we take the same species and tag them across multiple parts of the country, right? Do they do the same thing? Right? Or across species. So if we tag multiple species in the same area, are they solving the ecological challenges in the same way? I'll give you one example. This map here is a map that we generated to describe African elephants moving through Kruger National Park in South Africa. And the red lines are the tracks that elephants make, and these are elephant herds that we've collared and followed through the landscape. And what we find is that elephants make these repetitive loops to access water. Right? So they loop away to forage and loop back, and they loop back at the hottest time of day, right? In order to thermoregulate. This large mammal, the world's largest mammal, in fact, needs to access water to cool down. And so they shuttle back quickly to access water at the hottest time of day. And they make these loops through space and through time in order to get both food and to thermoregulate. Right? And we wouldn't know this if we didn't tag and track them across their landscape. Right? This takes us to the question, right? Where do we work? Right? Where does all the science that I just described happen? Our urban-rural gradient for all the questions that involve Psammophilus dorsalis, the rock agama, happen here in and around Bangalore city. And so the image I showed there is two adjacent habitat types. The urbanized landscape right up against rocky boulders, which is rural, unurbanized. So we are trying to understand the urban-rural context with one species, and that happens here. Our thermal and predation experiments or research program is currently in Desert National Park, and we're likely to expand that to Gujarat as well. Right? And finally, our multi-species comparisons of phenotypes, when we're comparing lizard species, or animal movement, when we're comparing mammals, happen all over India. So far, we've been anchored a little bit in the Western Ghats, but this is likely to expand across the entire country now. And none of the science that I just described would be possible without my lab. And so with great thanks and great enjoyment, here's the last picture we have of our lab, dated December 2019, before COVID-19 prevented us from coming back together. And so all the research program I describe here has been spurred by their own interests. And so it's bottom-up interest of our research. It's also been generously funded by all these funding agencies you see down below. So we're grateful to them as well. And with that, thank you very much. And it's time for me to head back to the field with my students. Bye-bye.